Dear students, today we will discuss the female reproductive structure in plants, which includes the pistil, the megasporangium, or the ovule, and the embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil that is monocarpellary or may have more than one pistil that is multicarpellary. When there is more than one, the pistils may be fused together, called syncarpous, or may be free, called apocarpous. Each pistil has three parts, that is, the stigma, style, and ovary. The stigma serves as a landing platform for pollen grain. The style is the elongated slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity or locule. The placenta is located inside the ovarian cavity. Arising from the placenta are the megasporangia, commonly called ovule. The number of ovules in an ovary may be one like wheat, paddy, mango, to many as in papaya, watermelon, orchids. Now let us see the structure of the ovule or the megasporangium. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus, except at the tip where a small opening, called the micropyle. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called a nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac, or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Now let us see what is megasporogenesis, the process of formation of megaspores. From the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell, or MMC, in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. Meiosis results in the production of 4 megaspores. That megaspore develops into female gametophyte. In a majority of flowering plants, one of the megaspores is functional, while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte or embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. Let us study about the formation of the embryo sac in detail. The nucleus of the functional megaspore divides methodically to form 2 nuclei, which move to the opposite poles, forming the 2-nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the formation of the 4-nucleate, and later the 8-nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. After the 8-nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte, or embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together, or at the micropylar end, and constitute the egg apparatus. The egg apparatus, in turn, consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip, called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalaza end and are called the antipodals. The large central cell has 2 polar nuclei. Thus, a typical angiosperm embryo sac, at maturity, though 8-nucleate, is 7-celled.